Hi all. Today we will see the metabolism of the glucose. So after digestion and absorption of the glucose, now the next step is the metabolism. Metabolism means metabolism, we know the catabolism plus anabolism. That is what we call the metabolism. So actually this carbohydrate metabolism, it involves a series of chemical reaction. So like glycogen formation, breakdown of glycogen and glucose to supply energy, formation of glucose from amino acids and glycerol or fat, and formation of fat from other are the main changes that related to the carbohydrate metabolism. So here we are given as in points like glycolysis, Krebs cycle, glycogenesis, glycogenolysis, and gluconeogenesis. So these are the various metabolism of glucose you are supposed to study. So here the glycolysis and the Krebs cycle are catabolic reaction, and glycogenesis and gluconeogenesis are considered as an anabolic reaction. Now we will see the glycolysis in detail. So normally in the glycolysis, there are totally 10 steps are involved. So first step is the glucose is converting into the glucose 6-phosphate. So here this is the first step, the phosphorylation of glucose to glucose 6-phosphate. So in this process, hexokinase glucokinase, that is the enzyme involved in this step, and one ATP. ATP is converted into, reduced into ADP. ATP means adenine triphosphate. So one ATP will enter into the reaction and liberated in the form of ADP, that is adenine diphosphate. So ATP is reduced to ATP here. Now the second step is this glucose 6-phosphate is converted into fructose 6-phosphate. So here the isomerization is taking place with the presence of enzyme phosphohexose isomerase. So this enzyme is involved in this particular step. Now the third step is fructose 6-phosphate is converted into fructose 1,6-biphosphate. So here is also the phosphorylation is taking place. So one ATP is reduced to ADP here, and the enzyme involved in the process is phosphofructokinase 1. Now this fructose 1,6-biphosphate undergo cleavage into forming two compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this fructose 1,6-phosphate to two compound, this is a reversible reaction. So this glyceraldehyde 3-phosphate can be forming dihydroxyacetone. So this reaction is also a reversible process. So fructose, during this cleavage process, the enzyme involved is aldolase. And here the conversion of a glyceraldehyde 3-phosphate to dihydroxyacetone phosphate is taking place the presence of the enzyme triose phosphate isomerase. Now this glyceraldehyde 3-phosphate is again undergo the further reaction. So here in the presence of enzyme glyceraldehyde 3-phosphate dehydrogenase, it forming a new compound called 1,3-biphosphoglycerate. So here one NAD is reduced to NADH, and the one NAD plus PI, that is inorganic phosphorus, is also entered into this particular reaction, and it released in the form of NADH and one hydrogen ion. Now, next step is 1,3-biphosphoglycerate is converted to 3-phosphoglycerate. So here, phosphoglycerate kinase is the enzyme here, and this particular step, the first ATP is formed here. The ADP is converted into ATP. Now, the 1,3-phosphoglycerate is forming to the 3-phosphoglycerate. Now, this compound is converted to forming 2-phosphoglycerate in the enzyme phosphoglycerate mutase. Now, the next 2-phosphoglycerate is converted into phosphoenol pyruvate in the presence of enzyme called enolase. And the last step, phosphoenol pyruvate is converted into pyruvate and in the presence of pyruvate kinase. So here also one ATP is formed. Okay, so this is the how the glycolysis process is taking place. So here total 10 steps are there. So you have to study this reaction like this, and you should also learn the name of enzymes involved in each reaction step, and also what all how many ATPs are produced here, what all things are liberated, what are things are added for that particular steps. So all those things you are supposed to study. Now this pyruvate will be converted into lactate in the condition of in the absence of oxygen. So, so this is the whole process of the glycolysis. Okay. So next is Krebs cycle or citric acid cycle or TCA cycle. So this is the major terminal pathway of the biological oxidation in all tissues. So normally the cycle is begin with the formation of a citric acid. So that's why it is called the citric acid cycle. And in this particular cycle, certain compounds are also contained some carboxylized group. So that's why it is called the tricarboxylic acid cycle or the TCA cycle. Okay, so now we will see how the Krebs cycle is happening. So in first step, the acetyl-CoA is combined with the oxaloacetate and it forming citric acid. So here the enzyme involved is citrate synthase, citric acid or citrate. Now the citric acid is converted to forming isocitric acid. The enzyme involved is aconitase. So in this particular step, no any energy molecules are coming or any water or carbon dioxide are also adding in this particular step. Now, the isocitric acid is converted into alpha-ketogluteric acid in the presence of isocitrate dehydrogenase. So here one carbon dioxide will be liberated here and one NAD molecule is reduced to forming NADH. Now, this alpha-ketogluteric acid by the release of one carbon dioxide and using of NAD to NADH, this is converted to forming the succinyl-CoA in the presence of hormone, not hormone, enzyme alpha-ketoglutarate dehydrogenase. Now this succinyl-CoA, remove this CoA and forming the succinate in the presence of enzyme succinyl-CoA synthase. So in this process, GDP is reduced to GTP. Now this succinate is converted to forming the fumarate in the presence of succinate dehydrogenase. So here the FAD is reduced to the FADH2. Now, by the addition of water, this fumarate is converted to forming malate and the presence of enzyme fumarase, and this malate converted to forming oxaloacetate by the formation of NADH and in the presence of enzyme called the malate dehydrogenase. So this oxaloacetate again combined with acetyl-CoA and forming the citric acid or citrate, and the cycle is continuing like this. So this is what we call the Krebs cycle or the citric acid cycle. Now we will see the next metabolism, glycogenesis. So from the name itself, it is clear that it is the formation or generation of the glycogen. We know the glucose is the simplest carbohydrate form, which is mainly used for the energy process. But after the requirement of the glucose for the body, the excess glucose will be converted or stored in the form of the glycogen. So that is explained in this process how this glycogen is forming. Normally, the glucose undergo phosphorylation and forming glucose 6-phosphate, and in the presence of enzyme called a phosphoglucomutase, this glucose 1-phosphate is again converted into UDP glucose in the presence of enzyme UDP glucose pyrophosphorylase. So here one UTP will enter into the process and liberated in the form of inorganic pyrophosphate, and this UDP glucose is converted to forming the glycogen. Okay, so that is done. That is taking place in the presence of enzyme called glycogen synthase, and one UDP molecule is also liberated here. So for the glycogenesis process, the starting molecules is of course the glucose. So then this whole process is called the glycogenesis. Okay. And that glycogen is stored in liver and the muscles. So that's all about the glycogenesis. Now we will see the glycogenolysis. So that is a biochemical pathway in which glycogen break down into glucose. Glycogenolysis means the splitting of that glycogen. So this glycogen will split into glucose 1-phosphate and the glucose. So this reaction is normally taking place in the liver and the muscles. So hepatocytes are the cells in the liver and mycocytes are the cells in the muscles. Now the whole process is under the regulation of two key enzymes called the phosphorylase kinase and the glycogen phosphorylase. we know the glucose is the main energy for whole action of our body. So during the fasting time, the glucose, we are not getting the glucose through the food. So what happens? So on that particular time, the liver will play a very important role. So that will converted the glycogen into the glucose. Glycogen, we know it is a branched polysaccharide which consists of lot of glucose units. Okay. So this glycogen will convert it into glucose, and if any need of glucose is there, automatically this glycogen will break down into the glucose. So the last one is gluconeogenesis. It is nothing, that is the synthesis of a new glucose from the non-carbohydrate precursor. So this is very important. In the last two university question, they repeated this question. What do you mean by the gluconeogenesis? So that is the formation of a glucose from the non-carbohydrate precursor. Okay. So it is also essential in the regulation of acid-base balance, amino acid metabolism, and the synthesis of carbohydrate-derived structural components. So the glucogenesis occurs normally in liver and the kidney. Now, the precursors of the gluconeogenesis are lactate, glycerol, amino acid with a propionate. So that is the what we call the non-carbohydrate precursor. So from this is the starting material for the formation of the glucose. So in this process, it consumes ATP for the energy purpose. So that ATP is normally formed by the oxidation of the fatty acids. So that's all about the gluconeogenesis. So this is the different types of metabolism of glucose you're supposed to study. Okay, thank you.